Our first speaker is Jennifer Moses, and she currently works for the Foundation of Applied Molecular Evolution, and she was actually a former biotech student of ours. So we're very pleased to have her. And then our second speaker is going to be CEO and President of Intermed. It's going to be Mr. Rick Staub, and I'll introduce him when his presentation is ready. I'm going to hand it over to Jennifer.
All right, well, I'm Jennifer Moses, and I'm a lab tech at Foundation for Applied Molecular Evolution, which is across the street in Progress Bar in Progress Center. There. If anybody wants to come visit me, you're more than welcome here. So a little bit of background about myself. I graduated from Keystone Heights High School, which is right outside of town. Yeah, I'm a local. In 1997, speaking back, I'm 36. I started taking college classes in the fall of 1999, sporadically part-time here and there. I've been on my own since I was 17, so taking classes here and there was what worked for me. So I finished an AA in nursing, which was all the prerequisites for nursing, from Santa Fe in 2010. And then I finished my AS in biotechnology here in 2012. I went straight to UF right after that and graduated UF last summer, 2014, with a bachelor's in biology and a minor in entomology. So approximately 15 years to finish my degree, which is great. Yeah, I finally did it. I was pretty excited about it. So, but the reason for that was I worked a lot. So I started off working at Sonny's Barbecue on Waldo Road, which is the original Sonny's, if you don't know that. Yes. Sonny's. And then after that, I moved up north and I became a receptionist and quickly learned that I was not receptionist material. I talked a lot. And it was way too outgoing for that. So I started working part-time in retail while I was taking classes at a community college up there. And then decided to quit being a receptionist, became an assistant manager, store manager, did the whole retail thing. Quit my retail job and decided to go back to school full-time. And that's when I decided to become a CNA. And then worked as a CNA for about a year and decided I didn't want to do patient care. And started working here. And that's where kind of like my lab career started. So I had taken all these prerequisites for nursing. I had taken, like I said, 2 years worth of classes for nursing. And then I got through and I was like, wait a minute, I don't want to do patient care. I became a CNA and I was like, I really don't want to do patient care. And so I was talking to one of my professors. I took anatomy and physiology at UF, Dr. Feldheim. Great professor. And she looked at me one day and she's like, I really don't think nursing is what you want to do. Because every project she ever gave me was one of those things that, anatomy and physiology is a lot of physiology. You're talking a lot about what's going on in the body. And that was more of what I was interested in. I wanted to do the physiology. I wanted to learn more about what was going on, the little details. So she kind of directed me, she said, you should go to a biotech open house. Learn about the biotech program. So I did. And that's where it all started. So I started classes here. I became a lab assistant here. And then I'll talk about the other two in just a minute. So at the Perry Center, when I was a lab assistant, I basically helped other students with their experiments. I think Jenny's son was probably one of the people I helped when I was here. And I cleaned a lot. A lot. I made a lot of reagents. I maintained all the cell lines that you guys do in the proteins classes. I checked all the equipment daily. I also tutored in anatomy and physiology for Brigitte, for her students, and for some of the students on the main campus. And then I got to start working on my own research projects while I was here, which was really what excited me. So the first one that I started working on. So, a little background on this is I went to visit my parents and back up north, moved back down there. I got a Scientific American magazine. And in there was this discussion about these silver nanoparticles. So this was kind of new way back then. And they were putting them in everything, but they weren't really regulated and they hadn't been studied that much. And they had started putting them in dryers as an antimicrobial. They were spraying them on the inside of the dryers. And the wastewater that was coming from these plants, from these factories, was getting into the streams and it was causing the fish in the streams to either die or the larvae or the fish, baby fish, born with mutations. And they would come out looking like the shapes of question marks and things like that. So already interesting me, and then I watched this talk online that they were going to make these silver nanoparticle water bottle filters that they were going to send to Africa and give all the people of Africa clean drinking water. And so my thought was that's probably not the best idea. And so I was going to do a little project just here while I was here, just to see if some of these filters might be, leaching these particles. I was curious, I didn't know. So I bought these 2 filters I found on Amazon that were advertising the silver nanoparticles, that it was antimicrobial one. That's, yada, 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 long story short. So I bought these filters, and I collected water and filtered everything, and I sent samples to UF. There was no silver in them. That's very disappointing. I'm not even sure there was actually silver in the filters. It's probably false advertising. But it was, like I said, a very good project for me. It was something I was really interested in. So it turned out to be just a fun thing to work on. So then I finished that, and Dr. Herko comes to me and says, how would you like to do a project on Alzheimer's? Like, that would be awesome. It would be so cool. Because as a CNA, I worked in the Alzheimer's unit at the village across from Santa Fe for about a year, and had worked on all of these patients, and of course it was really cool. So he says, have you ever heard of aptamers? I said, no, I have never heard of aptamers. So he came to me and explained to me these aptamers are these single-stranded pieces of DNA that'll fold and act like, and they'll bind to things. So we started this project, I guess I should say. I ended up getting an award, the Provost Research Award here at Santa Fe, to do the project. I presented my initial findings at the Root Festival on campus, and this project is still waiting to be finished. I believe my boxes are probably still in the freezer, waiting to be finished. So that was here at Santa Fe. So shortly after, I ended up going to get a lab tech job at UF. I was already going to be transferring to UF anyways, and starting a lab tech job over there was my next step in my career. Well, I got a job in the horticultural sciences building with Dr. Mark Sims, who does horticultural research. It was probably the hardest lab tech job I've ever had in my life. I did thousands and thousands of extractions on kernels, which if you've ever tried to extract DNA from a kernel, then you have to cut it with a scalpel. And you let it sit in water, and you beat it with a beater, and then the whole process. So I spent a lot of time doing that, thousands and thousands of extractions that go into thousands and thousands of PCRs. That go into, we grow the same ones up, and we find out if there's a phenotype that's matching the genotype, because that's important as well. But it was all genetic mapping, just to try to find out where the mutations were in the genome. That was my main job. I also learned how to use a tiller. I was sore for 3 days after I worked with tiller in 95-degree weather. So they are still, to this day, still finishing this project. That was 3 years ago. So they actually just finished the paper that I was on. They just submitted it. No idea if it's even been published yet. So yeah, 3 years, probably even longer than that in the making. So yeah. So one day, Mark came to me and said, how would you like to work at Foundation for Applied Molecular Evolution? My wife works there, they're looking for a lab tech. I needed a full-time position at the time. So I was like, absolutely. I already knew about the company because Matthew Kerrigan, that now works here and teaches biology. I had met him previously and he told me about the company. And Dr. Eric Herko actually told me originally about Dr. Benner and what they did. And so I'd already heard all about it. I was like, that was like my dream company to work for. So I've been there now for 3 years. My supervisor is Dr. Ryan Shaw, and the president of the company is Dr. Stephen Benner. So the main project that I really want is what's called AEGIS, which is Artificially Expanded Genetic Information System. You know that DNA consists of A and T and C and G. Well, Dr. Benner has decided that he wants to now incorporate all of these other binding patterns, all of these other, the P and Z, the S and B, K and X, J and B. I only work on the P and Z. So my talk is mainly going to be about that. And what it is alternative hydrogen bonding pattern. So they're only supposed to bind themselves, they're not supposed to bind an atom. So the P is supposed to bind the Z just like the T and the A and the C and the G. So that's what I'm working on right now. So as I they allowed me to take research credits and do a research project while I was at my work. So they gave me my own individual project to work on. And it was creating this POL1, which is DNA polymerase 125, and putting a mutation in that could accept the Z and the P and put the Z across from the P as it's writing. So I PCR'd out my gene, I made my mutations, I put it in the plasmid, I put it in my cell, I made my protein, I purified it, and I tested it. And this is how I tested it. This is what we call a 5' nuclease assay, but a lot of people call it TACMAN assay. We have modified it a little bit because we are dealing with C's and P's and normal stuff. So right here, so this is our template strand. So our template strand is what's going to have the P and the Z in it. So right here, this very first base after this primer is going to have a P or a Z. So what happens is your TAC or your polymerase, in my case, the polymerase 1 that I've mutated, comes in, finds that first base, puts their Z in across from the P, copies, hits this fluorescence, chews that off, sends off the fluorescence, and gives me a single signal. Now, if I don't put Z in there, across from my P, I get no signal, no fluorescence. Well, if I put wild type POL1 in there, which is a natural one that's expressed in E. coli all the time, that's these two here. So this is with and without DZ, so this dark blue and light blue. So it likes to misincorporate across from the P. It wants to put whatever it wants across from the P, because that's what POL1 does. POL1's a proofreader, so it goes in and it fixes things that aren't right. So it sees it as a mismatch, it fixes it, and it puts in whatever it thinks is supposed to be across from it. Well, my mutant, without Z, doesn't really do anything. If I put Z in it, I get this signal here. So my little experiment, I would say, was a success. It is a lot better at not misincorporating, and that was the goal of my project. So, I'm not done, I got more. So what I'm working on now, what my whole group is working on now, including Deeplin is here, she's part of my group, and she's helping us. So the main goal of this is to get these natural nucleosides, so DP, just DP without any phosphates, and DZ without any phosphates, get it to be taken up by the cell, get it from DP all the way to DPTP, so that we can get it into the DNA, and it'll just keep it, right? So in order to do that, we have to find these kinases that will take the DP or the DZ, put on a phosphate here, put on another phosphate, and then we have our triphosphate. So what I work on now is all trying to find these kinases that will do that. So we start with natural kinases, the ones that do regularly the A's, the C's, the T's, and the G's, trying to mutate them or find ones that, natural kinases that will work and that will do what we want them to do. And then try to find assays and things that we can trust to give us the answers that we want. So that's what I work on now. This is the main thing that I do every day. I take a gene, I put it in a plasmid, I grow it up, I induce it, I purify it, and I test it. And that is what I do every day.
I'm in Virtual Biotech, so I'm trying to understand what's the purpose of doing having extra nucleotides?
It's so funny because that's the first question I get every single time I talk about it. And I always tell the story. I told Erin the story before. So Dr. Benner is this very interesting person, and he always introduces us when he comes and brings people around. And he comes to my office and he's got the head of IDT, these people that create these logos. And he brings them into my office and he says, "This is Jennifer. She's trying to create a legally." And then she goes, and she collects bugs. And then he walks out, and that's what he does. So I'm not, honestly, I think we're just trying to get it to work. Whether or not to scream the alien would be really, really cool. But yeah, I mean, and if you think about what alien is, it's an alternative life form. And if you have 6 bases or 8 bases or 10, it's pretty alternative. So, yeah, that's pretty much the goal.
DNA from beyond.
Yes, well, when I first started the project, we were funded by DARPA. And so I was like, oh, we need super soldiers. And I realized that's wasn't what we were doing. But it would have been really cool. I was like, the Fort Identity Project. Thought that would have been really, really cool. But it wasn't. It wasn't anything, I think. So, yeah.
So you're still in the process of making sure that all this works before you audit anything. But do you have any ideas where you could try to apply just research based to see biology stuff?
I think it's really just to see where synthetic biology will take us. I mean, I think that's really the goal of it. I know that they have the other projects that's going on with the company, and they do a lot of diagnostics and things like that. I don't work on those projects, I can't speak for what they're using them for, but I would think that they could use them in detection assays and things like that. But my thought would be, if you could put in one of these alternative bases and people that have these diseases with overproductions of proteins and things like that, I mean, that would think you could be able to do tons of stuff that would work. Right now, we're just trying to get E. coli to do it, so see what happens. Anybody else?
They would not, if you put it there, they would not be able to translate.
No, most of the natural E. coli proteins will just completely override it, change it to whatever it wants to change it to. And we have shown that different strains with different knockouts will do different things. So it's depending on what proteins are being expressed in there is what it's going to change it to. So, I mean, most of the time they change it to their like substrates. So Z is like C, and P is like G, and usually mismatch accordingly with that. From my research. Anything else?
As a teacher of this program, okay, I want to know, how did you feel when you were out there in the real company? The experience that you had here as all of our students in the biotech.
Honestly, I love the program here, and I wish I could have done more while I was here. Honestly, I had no clue genetic mapping. I had no clue what I was doing, but they taught me, and I felt comfortable in my basic techniques. I knew how to pipette. I knew how to run jump. I knew how to set PCR. I knew those things from this program. But I think a lot of my curiosity and the knowledge that I got from doing my research projects is really what helped me get the jobs that I wanted to get. And people always tell me, like, you go and you work hard, you're going to get the job you want. Because they will see it. And I think that's what happened. Dr. Settles saw me. I was actually, at that time, I was not working in the lab. I was working 3 jobs. I was going through a divorce. And I was working at the mall, I'm still working at Dr. Settles' lab, and I was working at AutoZone, those 3 jobs. And Dr. Settles came back to me and he's like, Patricia is hiring. I already gave you a reference. You just got to talk to them. He's like, you should not be working like this. But you do what you got to do. I mean, I have a child. I was going through divorce at the time, so but I knew, like I said, if I worked hard, that I would get the right one. So yeah, so that's all I can tell you. Work hard.
Yeah, I would like to add something to that. I know Jennifer since we were both the students in the program. I've been a long time here, by the way. I'm a mom. I have like 3 kids.
Always, she was like, that was 4 years ago.
Yeah, but I said, that many years have passed so fast. Oh my God, I'm still here. But it's because, like she said, we have kids.
You do
that
where
are,
you can do it. 15
yeah,
years. It took me 15 years to get where I am right now.
Extracurricular activities. So where I'm going is that she was there in the lab. She was working and helping everybody, and that helps a lot. Doing it over and over and over again, repeating, asking questions, making mistakes, making lots of mistakes.
A lot of people for messing up their Western blots. I messed up my Western blot like twice, by the way.
that will, yeah, so.
But making the mistakes is what helps you learn in the future.
Yeah, and
That
asking.
my first project that I did at UF, but the one that I was creating the full one, I messed up on every single thing. And if I go back real quick, if I have a little bit of time. Okay. This right here. This is so far from the truth right now. So if you know anything about polymerase 1, it has this nuclease activity, right? If I put this into this mix, that normal TACMAN assay, it comes over here and it just clips off this fluorofluoride stuff. Doesn't help anything. It just takes out the fluorofluoride. I'm like, cool, I get signals for everything. So we realized that was really poor design on our part. And so we had to switch everything around. So first we tried this, where we put this here, we put this here, and it worked fine. It wasn't getting us very high signals. And then we realized, what if we just put the fluorescent here and the picture here, and then it worked perfectly. It's all beautiful now. But I mean, that was weeks and weeks and weeks of experiments trying to figure it out, and you had to wait, you had to order something new, and it had to come in, and it was expensive. And you had to try it, you had to try all the naturals all over again. All new templates, because the way this works is these templates, like I said, they have their P or their C, but say it was natural, say it had a C, and I was trying to see if it was putting a G across from it, right? Well, then I couldn't have any other C's in the rest of it. Well, then that means that every template is different, every probe is different, everything fits together like a puzzle. So not only did you have to just order a new template, you had to order probes and primers, everything was different. So yeah, so had I not failed miserably at everything the first time around, I wouldn't have learned as much as I learned. So I learned more from the failures than I did from the successes.
How do you know if it works to put those nucleotides? Did you have to sequence it? Because it showed that you did it STS data.
So how do you sequence something that has a non-natural base pair?
Right, that's what I'm asking.
Don't. So you have to run your negative controls. And your negative controls have to be negative. They have to be the flat line that you saw first in the next slide. You had to see no fluorescence in order to know that it was working.
So this is just a fluorescence on
Yes,
a,
this is measuring the fluorescence. And this is just one of the templates I actually had. 15 different templates that I
How
had.
long take you to finish that slide?
Do what?
How long take you to, to trial, so many times, the combination,
Well, when you're writing a paper, you have to do triplets to everything. So not only did I have to get it perfect, and I realized also while I was doing it that every template, whether it had a P or a Z in it, really liked a specific condition as well. So it wasn't just the template with the probe and primer, it was the buffer that it was in, how much enzyme it needed. It was a little bit different for every single one. So you have to optimize all of that before you can get anywhere near such a pretty picture of this.
signal.
So it took me probably a year to
So
get the
it's
results
like a
that way.
research laboratory, right? It's not?
Yes, this is a research lab, and it's very much an academic-based lab, which I'm very blessed in that I have such good teachers that I'm surrounded by. So I learn something new every single day that I'm there, every day.
Your president is genius.
He,
Yeah, Banner.
yes, yeah.
He can be genius.
He is.
Yeah.
He might be the next like kind of inside guy. He's a little bit out there, but so fun to be around. Like so interesting to hear him talk. And I've only had maybe 12 conversations with him, but he mostly talks at me. At me, but he'll ask me questions too, and it's questions that I have no idea what the answers are. Like the founder of some company. I'm like, yeah, so I'm supposed to learn this? So yeah, he's that type of person. He's very eccentric and very interesting, but he can come up with these ideas that you're just like, I never would have thought of that, just in a million years. So it helps to have somebody that's so well versed and educated that if you have a problem, you can just like, what's that? And you're like, oh, cool.
Do you mind how big is the company?
I'm sorry?
How big is the company now? How big is your company?
30 people?
Huh?
About. Give or take 35 people about. We have two different departments pretty much. We have a molecular biology, biochemistry side, and then we have the organic chemistry side. So we have, the organic chemistry side is all the synthesis of all the templates and nucleosides and everything that we would need. And they sell them to us, and we ranges them out, and then we test them.
Oh my God. It's so good.
Or what is your objective?
I believe that might have been why I defeated the test, but I am a lab tech, and I'm not privileged to a whole lot of information. But yeah, I think that my research project is funded by NSF. So we are, the side that I work on is nonprofit. So it's all research, all grant funded research. The other side, I honestly really don't know what they do. But they do sell, Firebird is the one that does all the marketing and selling of the products. So I just don't work with it. So.
I guess what I'm looking for is, what do you have an objective in mind when you do the research, or are you just doing the research?
So my project is theirs baby. So this is his life child, his thing that he wants to get done, and he gets it funded. How he can't. So this is his brainchild, and he just wants to be finished, so he just keep moving forward. The other stuff, like I said, he works on diagnostic stuff. So that's the other side of the. This is all just Bender's brain and actually what he wants us to do. That's what
So you guys do any evolution like he was doing a lot, you know?
I'm sorry, say that again?
I saw that he was collaborating with NASA or something.
I believe he used to do that. I'm not well versed in what his current collaborations are on my project. I stayed really busy though, so that's probably part of it.
They're different projects.
Oh,
Yeah,
okay.
yeah. There is an astrobiology
The one
line.
Lisa's doing?
Elisa, yeah.
Yeah.
I see it.
Yeah, I think they're doing more. I think that's what you're taking, the origins of life stuff.
Yeah,
Yes.
they're doing rocks. I know
Yeah,
some
rocks.
of the postdoc formal students are doing a rock. They even went to California to get a job or something.
You said you're in the park, you're in the supermarket.
The Progress Center. I believe we're affiliated with the incubator, though, correct? We're still one of their equals. We're able to go use the incubator facilities and things like that, so we're still part of them, but we have our own labs with them.
So you've been doing the organic chemistry, you've been doing this side for quite a while. Any speculation on how many nitrogenous bases can stand in for other nitrogenous bases? How many?
I have no idea.
Are
They have put, I think, 6
Okay.
into a template. So, and I know that is statement, I think they made a crystal structure of that, I think, was the latest news. They would have put 6 in. Yeah.
Thanks, Jennifer.